Okay, so in histology of pancreas, if you remember the histology of liver, what we have discussed, what I have shown you under microscope, okay, then I hope you can understand about the histology of pancreas. So pancreas is a which type of organ? Yes? No, that is okay, not related to the abdomen. In general, depending on the secretion, which type of organ it is? Endocrine and exocrine. Both endocrine as well as exocrine, both components are there in the pancreas. Okay, this is one point. Second point, same like the liver, pancreas is also covered by a connective tissue capsule. Okay, septa extending into the substance of the gland will divide the gland into numerous lobules. Okay, so you have to take one particular lobule and then you have to study. Along with the connective tissue, septa, blood vessels, nerves, lymphatics, they are also entering the gland. Okay, next, if you take particular one lobule, okay, you can see the secretory part. The majority of the secretory part in case of pancreas is made up of exocrine part. Okay, approximately 98% is made up of exocrine part. Only 2% is made up of endocrine part. Okay. Why I am telling you this? Because when you will see the slide, at that time you will come to know that there are some lobules where pancreatic exocrine part is present, but endocrine part is absent. Okay, so जो endocrine part होता है, it is distributed throughout the gland. If you see the endocrine part, it is distributed throughout the gland in between the exocrine part. Okay, but majority of them are situated in the tail of the pancreas, the endocrine part. Okay. And what is what does it called as the endocrine part? Islets. It is called as islets or islets of Langerhans. Okay, so this particular islets, the population of the islets are present most in the tail of the pancreas. So whenever you are writing in the exam or anywhere about the pancreas, especially the histology, you have to make it separate. First you have to write about the exocrine part, then about the endocrine part. Okay. Now in case of exocrine part, the cells will be different, the staining will be different. Okay, secretions will be different. Same in case of endocrine part also, cells will be different, staining different, secretions are different. Okay, so like this you have to study about the histology of pancreas. Okay, so pancreas, as I said, it is covered by a thin connective tissue capsule, okay, which will extend inward septa, partitioning glands into lobules. Now the parenchyma consists of pancreatic acini. Now this is very important. That means if you see the exocrine part, okay, it is mainly made up of pancreatic acini. Okay, along with the acini, there are a small amount of islets, okay, which are surrounded by stroma of loose connective tissue. That means mostly it is made up of pancreatic acini. Pancreatic acini is the part of exocrine part of the pancreas. Pancreatic acini are the exocrine part of the pancreas, okay, which secretes digestive enzymes. Next, you have to understand one thing. If the digestive enzymes are secreted by the pancreas, okay, how it will be conveyed, means how it will be conducted. Once it is secreted from one part to another, how it will go? With the help of ducts. Okay, so that means in case of pancreas also we are getting ducts. Different types of ducts are present inside the pancreas. Okay, so as you can see, interlobular connective tissue, septa project from the capsule into the pancreatic parenchyma, organizing into lobules. Now in this septa, the ducts are present. Okay, so the septa which is present, in that septa blood vessels, lymphatics, nerves, that will be there. Along with that, the ducts are also present. Okay, that is, different types of ducts are there. Okay, now those ducts are also important. Why? Because from the pancreatic acini, the secretion which will take place, mainly the enzymes, that has to be conducted. How it will be conducted? With the help of the ducts. Okay, so we have to study about the duct system also. Now always keep that in mind. Whenever you are studying about duct system, you have to know what are the different types of ducts present. They say which is the smallest duct, after that which is the next one, and which is the largest one. Okay, this is the duct pattern is situated in most of the glands. Next you have to understand, suppose the smallest duct, if it is there, what is the lining epithelium? Then what is the lining epithelium? And already I mentioned earlier that if you see the size of the duct as it enlarged, okay, from the smallest duct to the largest duct, as the size increases, size means the diameter of the duct, as it increases, the lining epithelium also changes. Okay, the lining epithelium also changes. So that also you have to understand in the duct system. Okay, so the pancreas is both exocrine, accessory digestive organ, and hormone secreting gland in our body. The exocrine pancreas is made up of compound tubuloalveolar gland. Okay, the exocrine pancreas is made up of compound tubuloalveolar gland. Okay, in that what happened? Pancreatic acini are present. So you have to understand one thing. Okay, so acini, when it is multiple, then it is called as acini. If it is one, then it is called as acinus. Okay, it is called as acinus. Next, if you see the acini, okay, from acini you have to take one particular acinus. Okay, so if you take one particular acinus, actually it is a type of gland, okay, where a small lumen is present. Okay, circular in section, lumen is there. तो जो secretions होता है from the compound tubuloalveolar gland, that will come into the lumen, and inside the gland, okay, inside the acini, cells are there, and those are called as acinar cells. Inside the acini, okay, what is present? Cells are there, acinar cells. From that acinar cells, secretions are taking place. Those secretion, where it will go? It will go into the lumen. Okay, so from that particular acini, acinar cells are there. From acinar cells, secretions will go into the lumen. Okay. Next, another important part is there. That is the duct. Okay, so arrangement कैसे होता है? Suppose this is the acini, okay, the duct will go into the center of the lumen of the acini. Okay, suppose this is one acinus, ठीक है, duct will go inside like this. So whatever secretions are there taking place from the acinar cells, it will come into the lumen, and from lumen it will go into the duct, and from duct it is coming out. ठीक है. Now, if you see the acinar cells, okay, if you see the acinar cells, they are almost like simple columnar epithelium with some modifications. Okay, they are almost like simple columnar epithelium with some modification. जो acinar cells होता है, okay, particular one acinus. Okay, so in case of exocrine part, as I said, pancreatic acini are there. Okay, now if you see the acini, they are made up of branched acinar gland, okay, which is surrounded by lobulated delicate connective tissue. Now if you see the interior of the acinus, stop talking. If you see the interior of the acinus, in that the lining epithelium which is present, okay, that is simple columnar epithelium. They are also called as acinus cell. But there are some modification. What is that modification? You can see cells having narrow luminal and broad basal surface. Okay. So the luminal part is narrow and basal part is broader. This is the modification of the acinar cell. Next, at the apical part of the cells, okay, zymosome granules are present. Okay, now those granules are very important because from those granules only the secretions are taking place. Next, with the help of H&E staining, this is very important because with the help of this you have to identify the cells. With the help of H&E staining, okay, if you see the cell, the basal part is basophilic and apical part is eosinophilic. Same cell, okay, basal part the stain is different, apical part stain is different. Basal part is basophilic, apical part is eosinophilic. इसका मतलब क्या है? What is the meaning of that? Say I am talking about the acinar cells. Now this is one particular acinus, okay, this is one particular acinus. Now these are the acinar cells, okay, ये पूरा एक acini है, ठीक है, one पूरा acinus है. Now in that the cells which you can see over here, what are this? These are the acinar cells, okay. Now in this particular acinar cells, if you see at the apical part, you can see the granular appearance. These are nothing but the zymosome granules from which the secretions are taking place, ठीक है. Next, you can see the position of the nucleus at the base of the cell. Now, when you will use H&E stain to stain this particular acinus, at that time what will happen? The basal part of the cell, will become basophilic and apical part of the cell will become eosinophilic. इसका मतलब क्या है? What is the meaning of this? हाँ, appearance क्या होगा? What will be the appearance? That means, if it is basophilic, then it will be blue in color or purple in color. Eosinophilic means it will be pink in color. Okay, so same cell, but in upper part and lower part, the staining difference will be there. From that particular point, we can identify the acinar cells. Okay? Why? Because can you see this is the duct, intercalated duct, which is going into the acinus. Okay, now this duct, it is also having cells. Okay, now as I have mentioned, if the duct is going inside like this into the acinus and you have taken the section, after that if you are preparing the slide, then what you will see? All around you will see the acinar cell, but at the center you will not see the acinar cell. You will see something different type of cells. Okay, so those are called as centroacinar cells. Centroacinar cells. Okay, so they are present at the center and these are the cells of the intercalated duct. Can you see the center of this acinus? क्या है center of the acinus में? Cells है, but which cells? These are the cells of the intercalated duct. Okay, and these are called as centroacinar cell. Now if you see the function of the centroacinar cell, they will produce bicarbonate ion. So after formation of the pancreatic juice, when it will go like this, when it will come out like this, okay, this particular centroacinar cell will secrete bicarbonate ion. So जो bicarbonate ion है, that will mix with the pancreatic juice and then it will leave like this, then it will come out. तो उससे क्या होगा? If bicarbonate ion is added to the pancreatic juice, it will become alkaline. Okay, pancreatic juice will become alkaline. That's why if you see the pH of the pancreatic juice, it is approximately 8.3, which is alkaline in nature. Okay, so the zymosome granules are there. Secretory granules contain digestive enzymes. Now this particular digestive enzymes which are secreted, mostly it contain protease, amylase, lipases and nuclease. Okay, these are the enzymes which are produced by this particular zymosome granules. Next, centroacinar cells. So I have shown you the centroacinar cell. Okay, these are located at the center of the acini. They represent the beginning of intercalated duct. Okay, that invaginate the acini. That means inward protrusion होता है. Now centroacinar cells show pale staining cytoplasm with centrally placed flattened nucleus. Okay. Now what is the function? Centroacinar cells secrete bicarbonate ions into the pancreatic juice. Okay, that is the main function of the centroacinar cells. Okay. Now centroacinar cells can also be identified under high magnification. Okay, and differentiation can also be done. Okay, how you can do the differentiation? Because it is like squamous cell. जो centroacinar cells होता है इसका appearance कैसे होता है? Just like squamous cell. And in case of acinar cell appearance कैसे होता है? Simple columnar epithelium. Okay, so from that difference it can be identified. Now this is the schematic diagram. Now in this particular schematic diagram, what you can see? First of all, you have to see the interlobular connective tissue septa. See, this is the interlobular connective tissue septa. Okay. ये वाला. This is the septa. This is the septa. Okay. Now what I have mentioned, that the connective tissue septa, what it will do? It will divide it into small lobules. So see, this is one lobule. This is one lobule. This is one lobule. This is one lobule. Okay, like this small lobules are there. Now take one particular lobule. Suppose you have taken this particular lobule. Okay, now what you can see in this lobule, majority is made up of pancreatic acini. ठीक है, तो ये देखो ये पूरा acini है. These are all pancreatic acini. Now among them you can see islets also. So these are the islets. So how we will differentiate between the pancreatic acini and the islets? Islets are pale staining. Pancreatic acini are dark stain. Under microscope, how we will identify? With the help of H&E staining. So whenever you will see the islet, जो islet होता है उसका appearance कैसे होता है? Pale staining. And जो acini होता है उसका appearance dark stain. Okay. So here you can see you have taken one particular lobule. So you can see these are the connective tissue septa. In the connective tissue septa, you can see blood vessels are present. Okay. After that, majority of them they are made up of pancreatic acini. Okay. And you can see pale staining group of cells which are present. Okay. And around that sheath of reticular fibers are also present. This is called as pancreatic islets. Okay. Now inside the pancreatic islets, you can see blood capillaries, fenestrated capillaries. Okay. Next, along with the pancreatic acini, you can see some ducts are also there. For example, you can see intercalated duct, and here you can see interlobular duct. How to identify intercalated interlobular duct? Based on the diameter, easily you can identify. Okay, just see the size of the interlobular duct. See the diameter; it is larger, and just see the intercalated duct. Now you have to note down at one point that those ducts are always associated with the pancreatic acini. जितना भी acini ducts है, those will be associated with the pancreatic acini. Okay, for example, see, these are pancreatic acini, these are acini. So along with that, here you are getting the duct. Okay, these all are acini. Here you are getting the duct. ठीक है, बट दिस इज आइलेट. आइलेट के अंदर एक भी डक्ट नहीं है. यू कांट सी एनी डक्ट बिकॉज व्हाई डक्ट्स आर नॉट प्रेजेंट? बिकॉज इट इज सिक्रेटिंग हार्मोन. हार्मोन विल गो टू द ब्लड डायरेक्टली. ओके, सो दैट पॉइंट ऑल्सो यू हैव टू आइडेंटिफाई अंडर माइक्रोस्कोप. Okay, next is the duct system. Now duct system the ducts which are present in the pancreas, first of all intercalated ducts are there, then we are getting the interlobular duct, and then main and accessory pancreatic duct. These are the different types of ducts which are present. So as I said, from smallest duct to the largest duct, first of all the diameter will be different, and along with that what will be different? The lining epithelium also. Okay, the lining epithelium is also different. Okay. Now beyond the interlobular duct, if you see the ducts, these are surrounded by loose connective tissue, septa containing smooth muscles and autonomic nerve fibers. Now the intercalated duct, okay, these are the interlobular duct, begin as centroacinar cells. So intercalated duct का beginning कहाँ पे होता है? Okay, at the centroacinar cells. Just now I have shown you from centroacinar cells the intercalated duct begins. Now if you see the lining epithelium, it may be simple squamous to very low cuboidal epithelium. ठीक है. Next, intercalated duct will open into interlobular duct. Now this interlobular duct are lined by cuboidal to low columnar epithelium. To see the epithelium is changing. Next, main accessory and pancreatic duct, these are lined by simple columnar epithelium. Okay, so see, first in case of intercalated we have seen it is simple squamous epithelium, okay, to low cuboidal. After that, in case of simple squamous to low cuboidal epithelium. Next, the interlobular duct, it is lined by cuboidal to low columnar, and the main pancreatic and accessory pancreatic duct lined by simple columnar epithelium. Okay, like this the epithelial lining also changes as the duct size of the duct increases. Now, intercalated duct का function already mentioned, bicarbonate ions and water in the pancreatic secretion. Now, ये जो secretions होता है, pancreatic secretions, mainly the exocrine part of the pancreas, it is controlled by what? जो pancreatic secretions होता है, I am talking about exocrine part, how it is controlled? It is controlled by hormones are also there, along with that autonomic nervous system is also playing an active role. Okay, so as you can see, it is under control of secretin and cholecystokinin hormones as well as the autonomic nervous system. Now, in this particular diagram, so what is this? One acinus. If it is one, then it is called as acinus. If it is multiple, then it is called as acini. Okay, so ये देखो, this is one acinus. Okay, next, inside the acinus, what are these? These are called as acinar cells. Okay, so what are these? These are called as acinar cells. Okay, now if you see the acinar cells, see the position of the nucleus at the base of the cell, and at the apex what you can see, zymosome granules. From these granules only the secretion of the enzymes are taking place. Okay, next, what is this? ये क्या है? What is this? This one. This is the duct, intercalated duct. As you can see, it is invaginating, means it is going into the center of the acinus. Okay. Next, the cells which will be present over here, the cells which are present over here, these are the centroacinar cells. Okay, these are the centroacinar cells. Now, what is the lining epithelium in case of centroacinar cell? Simple squamous to low cuboidal. Okay. Now, gradually the intercalated duct, as you can see, intercalated duct. After that, we are getting interlobular duct, then main and accessory pancreatic duct. Like this, the duct, as it is becoming larger, you can see the lining epithelium changes. Okay. Now, in this particular chart, you can see in the acini, first of all, simple columnar cells, basal basophilia, apical eosinophilia, as I have mentioned. Then centroacinar cell, simple squamous to low cuboidal epithelium. Okay. Next, in the intercalated duct, you can see low cuboidal epithelium. Then interlobular duct, cuboidal to low columnar. Okay. And main and accessory pancreatic duct, simple columnar epithelium. Like this, the duct pattern changes, the lining epithelium also changes. Okay, striated ducts are absent. Okay, striated ducts are present mainly in case of salivary glands. Another important type of cells which are present in the pancreas, these are called as pancreatic stellate cells. Okay, pancreatic stellate cells. Now, these particular pancreatic stellate cells, they are present at periacinar, periductal and perivascular spaces of the pancreas. These are some certain areas where these pancreatic stellate cells are present. Okay, so normal tissue architecture. Okay, this particular cell, they maintains the normal tissue architecture. How? They synthesis extracellular matrix protein, matrix degrading enzymes, okay, and tissue inhibitor metalloproteinases. Okay, by this it is maintaining the normal tissue architecture of the pancreas. Now, there are some cases where this particular cell is to get activated. Okay, when it will get activated, suppose oxidative stress is there, then interstitial pressure is there, hypoxia, hyperglycemia. Okay, during these cases, the cell is to get activated. Now, when the cell will get activated, at that time what will happen? At that time, various growth factor will be there to promote the growth and proliferation of the pancreatic cancer cell. Okay, so what it will do? It will promote the growth and proliferation of the pancreatic cancer cells, inhibit the apoptosis and enhance their invasion ability. Okay, so that is also an important part plays by the pancreatic stellate cells. Now here you can see the pancreatic stellate cells. Stellate cells means star-like appearance, as you can see in the diagram. Okay. Now pancreatic stellate cell cannot be seen under H&E staining. For that special type of staining method is required. Okay. After that only it can be seen. Normal H&E staining में दिखाई नहीं देता है. So immunohistochemical staining method is there, okay, where specific antibodies are used, okay, in order to stain this particular type of cells. Okay, so in the exocrine part, the pancreatic juice which is secreted, approximately 500 to 800 ml is secreted per day, and it is highly alkaline in nature. You can see the pH is 8 to 8.3. Now the pancreatic juice which is secreted, only enzymes are not there. Along with the enzyme, other substances are also present. Okay, as you can see, mostly it is water and some parts solids are also present. In the organic substances, you can see enzymes and other organic substances, and here you can see inorganic substances are also present. Okay. Now in biochemistry, I hope you have studied that whenever any enzyme is secreted, okay, that particular enzyme, it needs an activator, then it will act on the substance. After that, the byproduct will be there, right? So ये जो pancreatic enzymes है, you can see the name of the enzyme. Name of the enzyme activator is also there. Okay, then it will bind to a particular substance and then byproduct will be there. Okay, now that is all about the exocrine part of the pancreas. Okay, next is the endocrine part of the pancreas. Okay, so in endocrine part में क्या रहता है? What is present in the endocrine part of the pancreas? What is present in the endocrine part of the pancreas? Islets are also called as islets of Langerhans. So as I said, only 2% of the pancreatic tissue is made up of islets. Remaining entirely it is made up of exocrine part. ठीक है? Now endocrine part का function क्या है? It will produce hormones. Different types of hormones are produced. Okay. Next, if you see the pancreatic islets, they are distributed throughout the gland. Along with the pancreatic exocrine part, okay, they are distributed throughout the gland, but majority of them, they are present in the tail of the pancreas. Majority of the islets where they are situated at the tail of the pancreas. Now if you want to demarcate the islets from rest of the parenchyma, okay, first of all you will see a delicate sheath of reticular fibers are there. Okay, next, it will be pale in staining, delicate sheath of reticular fibers that will encircle the pancreatic islets. Then they will be pale in staining. Okay, and if you see the cells, spherical clusters of polygonal endocrine cells. Okay, spherical clusters of polygonal endocrine cells are there. Now on a histological slide with H&E, they appear as large pale staining cells enveloped by intensely staining basophilic pancreatic enzyme. Pancreatic acini, they are dark in stain, but they are light in stain. Okay. Next, if you see the cells of the islets, they are connected to each other. Now cells are not separated from each other. Okay, they are connected to each other with the help of desmosomes and gap junction. Okay. Next, pancreatic islets are permeated by fenestrated capillaries. That means fenestrated capillaries are present inside the pancreatic islets. That is required. Once the secretion of the hormone is taking place, it can go directly into the blood vessel. Okay, next, with the help of H&E staining, we can see only the pancreatic islets, but we cannot differentiate the cells, different types of cells. For that, what we have to do, special type of staining method is used, and that is called as Melroy-Azan staining, and the fixation method is called Zenkar fixation. Okay, so with the help of Zenkar's formal fixation, 3 different types of cells can be identified. In case of alpha cell, it is red in color, beta cell, it is brownish color, and for delta cell, it is blue in color. Okay, this is how the cells can be identified. Okay. Next, immunohistochemical staining are also there. With the help of that, we can identify the F cell or PP cell, D1 cell, and enterochromaffin cell. Okay, but specially for enterochromaffin cell, with the help of silver dichromate, okay, it can also be identified. With the help of silver salt, silver nitrate, these enterochromaffin cells can also be identified. Now, cells of the endocrine pancreas. So, what are the different cells present? As you can see, alpha cells, beta cells, delta cells. So function is glucagon, insulin, and somatostatin. For F or PP cells, pancreatic polypeptide D1 cell, vasoactive intestinal peptide, and enterochromaffin, they secrete serotonin, motilin, substance free, etc. Okay, these are the secretions. Okay. So please note that this part you have to mention in the practical notebook. Okay. So whenever you are writing, as I said, you have to make it separate, the exocrine part as well as the endocrine part. Okay. In endocrine part, what are the cells, particular secretion that you have to mention. Now, what is this? Anyone? What is this? This is the pancreatic islet. So as I said, if you see a particular pancreatic islet, first of all, it will be surrounded by delicate sheath of reticular fibers. Okay. As you can see, first of all, it will be covered by delicate sheath of reticular fibers. Okay. Next, you can see the cells which are present inside the pancreatic islets. They are attached to each other with the help of gap junction and desmosome. Next, you can see the fenestrated capillaries. Blood capillaries are present inside the islets. Okay, this is very important. And then you can see different types of cells which are present. Another important part which you have to know, that is the neuroinsular complex. Okay, neuroinsular complex. Now, what is this neuroinsular complex? Now, for that you have to understand the innervation of the pancreatic islets. So, the endocrine islets are mostly innervated by parasympathetic system. Okay, that means the parasympathetic system is controlling the secretion mostly. Now, fine branches will first ramify to form plexuses around the islets. Okay, then what will happen? First of all, the fibers will synapse with the acinar cells. Okay, then it will synapse with the cells of the endocrine part. Now, mainly fibers enter the islets along with the arterioles. So just now I have shown you the capillaries. Through that capillaries, nerve fibers, they will enter inside the islets. Okay, now parasympathetic ganglia lie in the connective tissue within and between the lobules, are frequently associated with islet cells, and they will form the neuroinsular complex. Okay, now let me show you the diagram. So what is this? This is a pancreatic islet. In the pancreatic islet, can you see different types of cells which are present? Color codes are given so that you can understand. Okay? See, different types of cells are there. Now, if you concentrate over here, you can see the ganglion cell. Okay. Now, these are the preganglionic fiber. These are the preganglionic fibers. Now, these preganglionic fibers will come and rely over here. From the ganglion, the fibers which are coming out, these are called as postganglionic fiber. Now, this you can see fibers are coming and then they are supplying the cells. Okay. One part. Another part, you can see the blood vessels are also entering the islets. So together, okay, this will form the neuroinsular complex, which is mainly controlling the secretion of the islets of the pancreas. Okay. Now, the secretion is controlled first of all mainly by the parasympathetic division, and along with that it is also controlled by the hormone secreted from pituitary gland. Okay, only not under parasympathetic control. It is under the hormonal control which is secreted by pituitary gland. Okay. Now, this is the histology of pancreas under low magnification. Now, what you can see over here, mostly what you can see, pancreatic acini are there. Now, in between the acini, can you see the pale areas? You can see the pale areas. So, what are these? These are pancreatic islets. So pancreas का जो slide है, identification is quite simple. It's not that difficult. ठीक है? Now let me show you in higher magnification. Yeah. So in higher magnification, ये सब क्या है? What are this? Okay. So this is the pancreatic acini. This is pancreatic acini. This is the islets. Okay. Now in the islets here, what you can see, these are the capillaries, fenestrate capillaries. This one, these are the capillaries. Okay. Next, can anyone tell me what is this? No, this one. ये देखो, ये part separate है. Not duct, connective tissue septa. I have told you, capsule से क्या निकलता है? Connective tissue septa, which will divide it into lobules. ठीक है? So these are the connective tissue septa. Okay. Now in the connective tissue septa, blood vessels will be there. Okay. Next, can you see the pancreatic acini? Okay. Now in the pancreatic acini, if you concentrate over one pancreatic acini, okay, you can see the zymosome granules also at the apical part of the cell. Next, you can see basal basophilia, you can see apical eosinophilia. That can also be identified under high magnification. Okay. Next, here ducts are not given properly, okay, but don't worry, in the slide I will show you the ducts also, intercalated duct and interlobular duct. Okay, so that is all about the histology of pancreas. Okay, suprarenal gland. So what do you think, which type of gland is this suprarenal gland? It is completely endocrine in nature. The suprarenal gland, it is completely endocrine in nature. Okay, it is secreting hormones. Now if it is endocrine in nature, that means there will be no ducts absent. Okay. Now today, during the lecture hour, have you understood anything about the structure of kidney, I have discussed, what I have explained? So first of all, what is present in the kidney? If you take a coronal section, क्या दिखाई देगा? Cortex and medulla. Okay, cortex and medulla. Now what is there in the cortex, what is there in the medulla, that we have discussed. In the same way, we have to study about the suprarenal glands also. That means if you take a suprarenal gland and if you take the LS of suprarenal gland, okay, if you have taken the suprarenal gland, if you have taken LS, you have opened the gland, ठीक है, इसके अंदर क्या दिखाई देगा? What you can see? You can see cortex and medulla. In case of suprarenal gland, the size of the cortex is larger. It is approximately 90% of the gland. Okay, 90% of the gland is occupied by cortex. Only 10% is made up of medulla. That means medulla is very small. Okay. Next, in case of cortex, different types of cells are present. Arrangements are different, and depending on the arrangement, the cortex is divided into 3 zones. Okay. From there, the various hormones are secreted. Okay. Next, when we will come to the medulla, at that time we will come to only 2 types of cells are there from which the secretions are taking place, and the size of the medulla is very small. Cortex is darkly stained, medulla is pale in stain. Okay, this is some important identifying feature of suprarenal gland. Now, so if you see here also same, you can see connective tissue capsule is there that will send septa inside the gland. Okay. Next, if you see the parenchyma of the gland, it is organized in 2 zones. That is the superficial cortex, 90%, deeper medulla, 10%. That means majority of it is made up of cortex. Okay. In the suprarenal cortex, it is arranged in the form of cords, and sinusoids are also present in between the cords. That means if you see the arrangement of the cells, these are in the form of cords. In between the cells, means in between the cords of the cells, what are present? Sinusoids are present. Okay. Here ducts are absent because entire gland is producing hormones. Okay, so those hormones, where it will go? It will go to the sinusoids. Now, if you can see the diagram, okay, to see the suprarenal gland, once the section is taken, after that you can see the larger part is the cortex, this smaller part is the medulla. Okay, so outer part first you will be getting the connective tissue capsule. Below the capsule, this entire part is made up of cortex. Okay, so 3 zones are there. You can see this is the outermost zone, this is the middle or the largest zone, and this is the innermost or the smallest zone of the cortex. Now in this particular diagram you can see the arrangement of the cells are different. In this particular zone it is different, in this zone it is different, and in this zone the arrangement of the cells are different. and in between the cells what are present, sinusoids are there, so that the secretion can come directly into the blood. Okay, and after that you can see the small medulla. So only 10% is made up of medulla, remaining 90% is made up of cortex. Now on the basis of the arrangement of the cell, the cortex is divided into 3 zones. What are these 3 zones? Zona glomerulosa, fasciculata and zona reticularis. 3 zones are there. Okay? Now among them, glomerulosa is taking 15%, fasciculata is occupying middle 80%, and then reticularis is inner 5%. Now if you see the suprarenal gland, okay, mainly the cortex, they are steroid secreting cells, that they do not store the secretory product, but they release it whenever it is required. So first is the zona glomerulosa, how the cells are arranged in case of zona glomerulosa. First of all, outer 1/5 of the cortex is occupied by zona glomerulosa. Okay, how the cells are arranged? Inverted U-shaped. Okay, they are arranged in inverted U-shaped cords and round like acinus group surrounded by capillaries. This is the arrangement of the cells present in zona glomerulosa. Okay. Next, with the help of light microscope, if you see zona glomerulosa, okay, small polyhedral or columnar cells are there with basophilic cytoplasm and deeply stained nuclei. These are the shape of the cells present in zona glomerulosa. Okay, polyhedral or columnar. Now, what is the function of these particular cells? They secrete mineralocorticoid hormone. What is the mineralocorticoid hormone? Aldosterone or deoxycorticosterone. इसका फंक्शन क्या है? It maintains the electrolyte and water balance of the body. Okay. Next is the zona fasciculata. Now in case of zona fasciculata, what is the arrangement of the cells? They are in the form of straight columns, 2 cell thick. That means 2 layers of cell will form 1 column, then another column. In between both the column, 1 small space is there which is occupied by the capillaries, blood capillaries. Okay, this is the arrangement in case of zona fasciculata. Okay, now zona fasciculata is occupying 3/5 of the cortex. Zona fasciculata कहाँ पे रहता है? Middle में रहता है. Okay, most of the cortex is made up of zona fasciculata, approximately 80%. ठीक है? Now in case of zona fasciculata, these particular cells, they are lipid rich, means lipid droplets are present. Whenever processing is done, those lipid droplets are washed away. Okay, they appear vacuolated. That's why they are also called as spongiocytes. Okay. Next, if you see the zona fasciculata, after that zona reticularis, इसमें arrangement of cells कैसे रहता है? How the cells are arranged? In the form of branching pattern, network like structure. Zona reticularis में जो cells होता है, how they are arranged? In the form of network. Okay, and in between them the capillaries are present. Okay. Okay, the fasciculata, already I have told you what is the arrangement. Now in case of fasciculata, the hormones which are secreted, these are cortisol and cortisol. Okay, now they are having decreased antibody response and have important anti-inflammatory effect. In case of reticularis, as I said, how the cells are arranged in the form of reticulum or network. Okay, and then they are separated by fenestrated capillaries. Okay, now in case of zona reticularis, these cells are smaller as compared to the zona fasciculata. Okay, now the cells are darkly stained nuclei, well developed, smooth ER and rough ER are also present. Now these particular cells, they are having a yellow brown pigment. This is called as lipofuscin granules, which are present in the cells. What is the function? This particular cell secrete weak androgen, which are precursor of what? Yes, precursor of sex hormones. Now this is the schematic diagram. Okay, now in this particular schematic diagram, first of all you can see this is the capsule. ये देखो, this is the capsule. Below the capsule, this particular part is zona glomerulosa. This large part, okay, this large part, it is made up of zona fasciculata, and this part is zona reticularis. Okay, now can you see the empty spaces? So these are nothing but the capillaries, okay, and then this is small medulla. Now here you can see the arrangement. In case of zona glomerulosa, inverted U-shaped clusters of cell. In case of fasciculata, you can see the cells are arranged in the form of columns, okay, separated by capillaries. And in case of reticularis, it is in the form of network, and in between them the fenestrated capillaries are present, okay. Now cortex is done, then this particular part, medulla, is left. So what is present in the medulla? Okay, so medulla is functionally, structurally, embryologically, it is different than the cortex. Okay, mainly two types of cells are there. As you can see, chromaffin cells are there and sympathetic ganglion cells are there. Okay. Now, if you see the chromaffin cells, why they are called as chromaffin cell? Because they take a stain that is called as potassium dichromate. That's why it is called as chromaffin cells. Okay. Now chromaffin cells जो होता है, if you see, they are columnar or polyhedral in shape. And what is the function of this chromaffin cell? They secretes adrenaline or noradrenaline. Okay, and this particular sympathetic ganglion cell, okay, they control the secretion from the cortex. This is the function of the sympathetic ganglion cells. Now this chromaffin cell, they secretes adrenaline and noradrenaline into the blood, okay, and I hope you know what is the function of the adrenaline and noradrenaline in the blood, okay. Now these are the different zones, okay. Now how you can identify or how you can differentiate the cortex and the medulla? First of all, in case of cortex, it is darkly stained, medulla is in pale stain. Okay? In case of medulla, if you see the sinusoids, they are larger, or you can say they are wider as compared to the sinusoids which are present in the cortex. Okay? This is the way you can identify. Next, in case of cortex, as I said, two types of cells are there. That is, chromaffin cells are there and sympathetic ganglion cells are there. Okay. Now the shape of the sympathetic ganglion cell and the chromaffin cells are also different. Okay. In case of sympathetic cells, if you see the shape, it is rounded or polygonal in shape. Whereas in case of the chromaffin cells, what is the shape? It is polyhedral in shape. Okay, so like this, depending on the shape of the cell, it can be identified. Okay, so now we'll go to the lab and then we'll identify the slides. First we'll identify the slides of pancreas, followed by the suprarenal gland.